Good afternoon and welcome to the North American Vascular Biology Organization's second webinar. Today's webinar is sponsored by DMT. I'm Linda Shapiro from the University of Connecticut, and we're pleased to welcome our speaker, Jessica Wagenseil, who will present her work entitled Elastic Fibers and the Mechanics of Maturing Arteries. Today's talk will be a more in-depth version of her presentation on the vascular mechanics session at Vascular Biology 2017 in Monterey this past October. Much of the presentation will refer to her article in the Journal of Biomechanics. Cross-linked elastic fibers are necessary for low energy loss in the ascending aorta. A link to the abstract of the article is included in the program materials. If you have a subscription to the journal, you'll have access to the full text. Throughout the webinar, you'll be able to switch between the phone audio and computer audio in case you're having problems. You can see this information on the audio section of the GoToWebinar control panel. If you experience technical problems, please click on the Help tab at the top of the control panel. Scroll to the bottom of the help screen for the technical support phone number. I'd also like to welcome Austin Cochalone, also from Washington University, who will monitor today's questions. Questions will be handled in two ways. Throughout the presentation, you could type your questions into the question box on the control panel. Your questions will be answered at the end of the presentation. At the end of the question and answer period, provided there's time, if you have another question, please raise your hand by clicking on the hand icon on the left-hand side of your panel. I will recognize you and your mic will be unmuted. You will then be able to ask your question live. This webinar is being recorded and archived on the NAVBO website for future use. Dr. Wagenseil is a biomedical engineer who studies the mechanics of large arteries and development disease. She's specifically interested in diseases that affect the matrix proteins involved in elastic fiber assembly. She uses mice with elastic fiber defects to understand the interplay between matrix protein amounts and organization, arterial mechanics, hemodynamics, and cardiovascular function. She also uses mathematical models to predict arterial mechanics based on changes to the matrix proteins and the hemodynamic stimuli. Please welcome Dr. Wagenseil.
Thank you, Linda. Today I will present elastic fibers and the mechanics of maturing arteries. The arteries that I focus on include the ascending aorta, which is shown here in this video, and then the descending aorta, which is shown down at the bottom. And what's important to note here is you can see the aortic valve opening during systole as the heart ejects blood. So it will open right now. Blood comes out and the aortic wall expands. During diastole, when no blood is being pumped out, the aortic wall slowly relaxes, which pushes blood further downstream and reduces pulsatile blood flow and pressure throughout the distal arteries. And the aortic wall is made up of a specific combination of extracellular or ECM proteins that allow this elastic relaxation. The matrix proteins that provide the mechanical behavior for the passive artery are collagen, which is highlighted here in green in this fluorescent micrograph of a mouse aorta. You can see most of the collagen is out here in the adventitia marked with an A. And then in the media are all of these red layers, which are the elastin and elastic fibers. They're organized in these concentric layers with some collagen associated with them. And then cells, which you can see marked with the cyan, are in between the layers with the intima at the bottom of the image. These elastic fibers are assembled in a very spatially and temporally regulated fashion that we still don't fully understand. There are two components of the elastic fibers. There's a microfibrillar scaffold that involves proteins such as fibronectin, fibrillin 1, fibrillin 2. And then there's the elastin core, which starts with secretion of tropoelastin, which is the soluble version of the elastin protein. The tropoelastin coacervates or self-associates, and then it matures. And fibrillin 4 and fibrillin 5 play a role in these steps. The elastin coacervates are then cross-linked by lysyl oxidase and lysyl oxidase like 1, and fibrillin 4 and fibrillin 5 also play a role in this step. And then they're finally assembled by depositing onto the microfibrils. And the proteins that I'm going to focus on today are 3 of the proteins involved in this assembly process, which are tropoelastin or elastin, fibrillin 4, and lysyl oxidase. There are mouse models with knockouts of all of these 3 proteins, and in all 3 cases, the mice die at birth with severe cardiovascular abnormalities. This is an image from the elastin knockout mouse, and it lives a few hours after birth, about a day. And you can see in this micrograph at postnatal day 0.5, the smooth muscle cells overproliferate, and eventually they occlude the vessel lumen. In the lysyl oxidase knockout mouse, these mice have large aneurysms, which you can see here. And then in the fibrillin 4 knockout mice, these mice also get aneurysms, and you can see in this micrograph where the elastic fibers are stained in black that they have very little assembled elastic fibers, and so they're missing in a large part of the wall. So for all 3 of these mice, we are looking at major components of the elastic fibers, and either the elastin core or the cross-linking or the assembly and cross-linking. In all 3 cases, the cardiovascular phenotypes include smooth muscle cell proliferation, which you can see here. It also includes tortuosity. And then for the elastin knockout mice, we see aortic stenosis with the smooth muscle cells occluding the vessel wall, or the lumen. And then in the lysyl oxidase knockout and the fibrillin 4 knockout, we see aortic aneurysms. And in all 3 of these cases, these are linked to human cardiovascular phenotypes associated with these genes as well. And so the goal of our research here is to quantify mechanical changes in the aortic wall that accompany severe elastic fiber defects and may contribute to the disease progression. And we also wanted to compare and contrast how the absence of each protein affects aortic mechanics and learn more about their role in the composite aortic wall and in elastic fibers. We started first by looking at some morphology. So we inject yellow latex in through the left ventricle and to circulate it into the systemic vessels. We remove the heart in most of these pictures, except for this one right here still has the heart. We have the ascending aorta, the descending aorta, and then the carotid arteries are marked as well. These are all newborn mice that are captured before the knockouts die. So you can see in the wild type, we have a nice straight ascending and descending aorta. In the elastin knockout, we have these stenoses that we see in the ascending aorta. You can also see some here in the descending aorta. And we have severe tortuosity of the carotid arteries, and then also down here in the descending aorta. In the lysyl oxidase knockout animal, we also have the tortuosity in the descending aorta. This is the ascending aorta here. About 30% of the lysyl oxidase knockout animals have patent ductus arteriosus, which you can see here. And these ascending aortas do dilate, although not in all cases, and it's not as often obvious from these pictures. In the fibrillin 4 knockout mice, we have tortuosity again, we have the descending aorta, and then we have a dilation of the ascending aorta. We looked at the ultrastructure of the wall using fluorescence microscopy. So in all of these pictures, the intima and the lumen are down at the bottom. So the top panel shows elastic fibers and elastin. So you can see these nice layers in the wild type mice. They're completely absent in the elastin knockout mice. And in both the lysyl oxidase and fibrillin 4 knockout mice, they're highly fragmented. The collagen fibers are not obviously different in the 3 cases. And then for the cellular organization, what we see is a lot of disorganization, especially at the luminal area for the cells. We have additional layers of cells across the wall, thickening of the wall, and then they're not oriented in the circumferential direction toward the luminal side. And you can see that in this composite image shown here. We wanted to look at the protein amounts to confirm what we would expect when we're missing these genes. So what we're showing here is elastin normalized by total protein, and we're measuring desmosine, which is an elastin-specific crosslink. So this is specifically for cross-linked elastin. So in the elastin knockout aorta, we have no elastin crosslinks, as one would expect. And then in the lysyl oxidase knockout and fibulin 4 knockout, we have large decreases, about 60% for the lysyl oxidase knockout and about 80% for the fibulin 4 knockout. We measured collagen as hydroxyproline normalized by total protein amounts. And so total collagen amounts are not different between these different mouse models. So even though we have severe defects in the elastic fibers, we don't see a compensation in total collagen amounts. We also wanted to look at the geometry of the vessel, and this is looking at the unloaded diameter. We do see a slight decrease in the elastin knockouts, and then we have an increase in the lysyl oxidase knockout and the fibulin 4 knockout, consistent with the stenosis phenotype you see in the elastins and the aneurysm phenotype that you see in the other two genotypes. When we look at the wall thickness, you can see significant wall thickening across all three knockout models, consistent with our microscopy results, where you see these extra layers of cells and thickening of the wall, and that's significant in all three of the models. So we wanted to look at mechanical behavior of the wall, and to do that, we use a pressure myograph from DMT. So we mount the vessel in a system and put it on a microscope that allows us to visualize the diameter, and then the system controls the pressure so we can visualize diameters at different pressures. One of the key components is that we use custom-made stainless steel cannulae so that we can actually secure the vessel tightly, and then we can stretch it in the axial direction while inflating it in the circumferential direction. And so we can control these two different directions to reproduce the physiologic loading in the mouse. And in the first set of results I'm going to show you on these knockout animals, I'm going to show you all data for the ascending aorta, which is defined here. When we do our pressure diameter tests on the myograph, you can see that the elastin knockout aorta starts at a smaller diameter than the wild type here in black. And what you're seeing in these curves is the dashed line is always the loading curve. So we pressurize it up to 60 millimeters of mercury, which is about double what you would expect the physiologic pressure to be in a newborn mouse. And then we unload it, which is the solid curve that you're seeing in both cases. And so the elastin knockout is always shifted to the left and a smaller diameter than the wild type. And then the other thing that's important is the difference between the loading and the unloading. So for a wild type, they follow more or less the same path, shifted slightly. But in the knockouts, they follow a very different path with loading and unloading, which I'll talk about more a little bit later. For the lysyl oxidase knockout animals, now we have this shift to larger diameters. And they also have a larger difference in the loading and unloading paths. And then the fibulin 4 knockouts also have a shift to larger diameters than their wild type, with again, a larger difference between these two paths. And in the lysyl oxidase knockout, note that the starting diameter was actually not bigger than the wild type, which is consistent with those morphology pictures where we couldn't really see the aneurysms. But once you inflate it and pressurize it, then you see this large increase in diameter, similar to the fibulin 4 knockout, consistent with the aneurysm phenotype. So those were pressure and diameter curves, which tell us structural mechanical properties, but we're interested in the material properties and how the lack of these proteins changes the actual material of the wall. And so we convert our diameters, which are shown here as loaded diameters over unloaded, into stretch ratios. And then we convert the pressure information along with diameters into stresses. And then we further take those to one more step where we're actually calculating green strain versus second Piola-Kirchhoff stress, because these two measures are energy conjugates. And so we can look at the energy that's stored or dissipated in the wall during our cyclic loading. And we're making some assumptions here in that there's no shear strains on the vessel and that we're looking at radial variations across the wall from the inside to the outside. So now if we look at stress versus strain, you can see quite a different behavior than what we saw with the pressure versus diameter. So now the elastin knockout is actually shifted to higher strains and lower stresses compared to the wild type. And then for the lysyl oxidase and for the fibrillin 4, we don't see as large of a differences in the stress and strain behavior as we did with the pressure diameter behavior. And there are some differences, but more or less these curves lay on top of the wild type. What is maintained is this large difference between the loading and the unloading in each case for the knockout compared to its wild type. In order to quantify our stress and strain behavior, we did some simple fitting where we fit a bilinear equation, one straight line that meets another straight line, using least squares fit to the data so that we can estimate the low modulus or the stiffness of the aorta at low strains and the high modulus or the material stiffness of the aorta at high strains. And then as well as this point where it shifts. And so we call that the transition strain and the transition stress. And this type of fitting has been done a lot in the soft tissue literature. And typically, people attribute this low strain behavior and this low modulus to the elastin contribution, and then this high modulus behavior to the collagen contribution. And so we thought this would be a good way to look at how these contributions change when we are lacking these 3 proteins. So when we look at the transition strain, so remember this is how far you strain it before it starts to become stiff and the collagen kicks in. And that really doesn't change across any of the 3 models for wild type in black and knockout in gray, or for the loading versus unloading, where the open symbols are all of the loading and the closed symbols are all of the unloading. What does change, however, for almost all of the groups, except this LOX loading case right here, is the transition stress. And so while it's being stretched to the same strain, the stress at which the aorta really starts to stiffen is much lower in these models where we have these elastic fiber defects. If we look at the modulus, this low modulus that is attributed to the contributions of elastin is much lower in the elastin knockout mice. But surprisingly, when you have elastin but it's not formed or cross-linked properly, it does not have a significant effect on this low modulus. If we look at the high modulus, which is where the collagen should be contributing, we don't have any significant changes across any of the models. The other thing we wanted to quantify was this idea of differences between the loading, which are the triangles right here, and the unloading, which are the circles. And so when we plot our stress and strain in this way, we can calculate the energy that's stored in the vessel wall due to the deformation by taking the area under this curve. So the stored energy when you stretch the vessel to this strain is the dark gray plus the light gray. And then when you unload it, if it were a perfectly elastic rubber band, it would follow the same curve back down. So you would get all of the energy back returned. But then whatever's dissipated by something like viscous losses or heat losses is this light gray curve and is the dissipated energy. And so I pointed out that there were large differences between loading and unloading in the knockouts. So we wanted to quantify these energy values. If we look at the energy stored, so how much energy is stored in the wall by the deformation, it's about the same across all of our models. However, the returned energy is much lower in all of the 3 knockout cases. And this leads to a percent dissipated energy, or basically the area between the 2 curves, that's around 70% in the elastin knockout and about 50% in the lysyl oxidase knockout and fibrillin 4 knockouts, compared to somewhere 10 to 20% for the wild type aortas. Now, dissipated energy in an adult aorta is usually about 8%. So we do have a higher dissipated energy in a neonatal mouse aorta, which is consistent with the idea that the elastic fibers are not completely formed at that point. And then when we have severe defects in those elastic fibers, we have a very large increase in dissipated energy. And so what we found by looking at these mechanical properties of these 3 different mouse models of severe elastic defects are that the consistent changes are this reduction in the transition stress and this large increase in the dissipated energy. And so why does that matter? I'd like to point out for the transition stress, this decrease is represented here, where this is where these 2 curves would meet for a wild type, and then for the knockout, the transition stress is a lot lower. And I mentioned that it's traditionally been attributed that this part of the curve is due to the elastin, and this part of the curve is due to the collagen. But most of those models are for adult aortas, where you're looking at an aorta with lots and lots of matrix and that the cells don't contribute much passively. But the newborn aorta has less matrix and the cells likely contribute more. And so what we hypothesize is that this shift to where elastin cannot support the stresses up to this level may have a large impact on the stresses that the cells are actually feeling, and that may change the mechanical response of the cells. For the dissipated energy, so the large amount of dissipated energy is consistent with a viscoelastic matrix rather than an elastic matrix. So it's not a perfect rubber band that returns all of the energy. It's a rubber band that has viscous losses or like fluid dissipation. And there's been some recent work looking at how viscoelastic forces affect cellular phenotypes. So you may be familiar with changes in elasticity, where people look at growing cells on materials with different stiffnesses and how the elasticity of that matrix or the modulus can change things like stem cell differentiation or cellular behavior. But now people are realizing that the viscoelastic properties as well, in this case it was changing stem cell migration and proliferation. And the hypothesis is that this time-dependent relaxation may allow matrix reorganization that can lead to ligand clustering and change the cell behavior. So we've talked a lot about mechanics, and I've implicated that those mechanics may change cellular signaling. So what about biochemical signaling? For example, how does the loss of elastin, fibrillin 4, or lysyl oxidase and the resulting changes in mechanical behavior affect things like gene expression? And we were also wondering if there's a global response to severe elastic fiber defects, and if there were any differences in the gene expression between the aneurysmal phenotypes that we see in the fibrillin 4 and lysyl oxidase knockout animals and the non-aneurysmal phenotypes that we see in the elastin knockouts. So to start to address some of these questions, we did a gene array study, and we used our 3 different genetic models. We had our 2 genotypes, wild type and knockout. We looked at 2 different developmental stages, embryonic day 17.5, which is about 3 days before birth, and then postnatal day 0. And then we looked at 2 vascular locations, the ascending aorta and the descending aorta. Because these are very small segments in newborn mice, we had to pool 8 segments per group, and then we did 2 samples of pooled segments per groups, which is a total number of data sets for 48 for these combinations, and then that involved 384 mice, and we used the Affymetrix Mouse 2.0 array platform. And we started this several years ago, and then we wanted to keep the same platform as we collected more data. We wanted to do these 2 different stages because we've previously seen large changes between this late embryonic stage, in this case it's marked as E18, to postnatal day 1. This is the systolic blood pressure measured in a wild type and elastin knockout mouse. They're about the same at embryonic day 18, and then we have a large increase in the elastin knockout at birth. We also see large changes in the morphology, where here's a wild type at E18 and an elastin knockout at E18. This is the ascending aorta. You can see it's longer here, but it doesn't have those severe morphologic defects that we see later at P1, including the stenoses and then the large hematomas. We also see large changes in the wall between these two time points, where here's an elastin wild type, an electron micrograph. The black are the elastic fibers. And then in an E18 elastin knockout, there's no elastin, but the cells are still organized in these layers. And then by postnatal day 1, the cells have started to overproliferate and become disorganized. So we really wanted to capture changes before and after some of these large mechanical and morphological and structural changes. And the reason we wanted to capture these two locations is because there are lots of cases where diseases depend on vascular location. And then specifically for the fibrillin 4 model and the lysyl oxidase model, we had seen before that this aneurysm phenotype, or this large increase in diameter for the fibrillin 4 knockout and the lysyl oxidase knockout, is specific to the ascending aorta. And so down here in the dotted lines, you can see the descending aorta for both the wild type and the knockouts, and those are quite similar. So we wanted to look for changes that occurred regardless of the stage and the vascular location, and look at how those two things interacted with the elastic fiber defects. For the analyses, we did some individual gene analyses where we looked at each genetic model. We ran each model on the same gene chip, and so we only compared within models to avoid batch effects. We did a 3-way ANOVA to look for changes in genotype, developmental stage, and vascular location, and then generated lists of gene changes if the fold change was greater than 2 and a p-value less than 0.05. We also did some gene set enrichment analyses where we looked at enrichment of Hallmark and Canonical gene sets from the molecular signatures database using the GSEA software that you can download at that site. We generated lists for p less than 0.05 and false discovery rates of 0.25 for the Hallmark set or 0.1 for the Canonical set. So I won't go into a lot of detail because we're just starting these analyses, but this is a Venn diagram of differentially regulated genes for fibrillin 4 model in orange, the elastin model in green, and the lysyl oxidase model in purple for wild type versus knockout. So every gene here was in the 3-way ANOVA was significant for upregulation or differential regulation in the wild type versus knockout. Some are up, some are down. And the only 3 that overlap between all 3 models include collagen 8 alpha 1, IGF BP2, and then thrombospondin 1. All of the rest, there is some overlap between 2 of the 3 models, and then there are some that are specific to those models only. The genes, so elastin, EFMP2 here is the gene for fibrillin 4, and then lysyl oxidase are differentially regulated just within those models. So it's interesting that none of the other traditional elastic fiber genes are differentially regulated when you knock out one of these 3. We also do not see upregulation of fibrillar collagens that you might expect to contribute to mechanics of the wall when you have defects in the elastic fibers. So I'll just briefly mention these 3 and what we know about these genes so far. Collagen 8 alpha 1 is a short chain collagen expressed by vascular endothelial cells. It's upregulated in vascular injury and disease. It promotes smooth muscle cell migration, and it associates with microfibrils in some but not all elastic fibers. For IGF-BP2, it's an IGF binding protein in the plasma that's expressed by vascular endothelial cells. It enhances IGF-induced smooth muscle cell migration and proliferation. And then thrombospondin 1 associates with the ECM and is expressed by fibroblasts. It modulates cell adhesion, proliferation, TGF-beta activity, and signaling from inflammatory cells. So these 3 genes are expressed by either endothelial cells or fibroblasts, but they do have large effects on smooth muscle cell migration and proliferation, so they may contribute to that additional wall thickening and cell layers that we see. And they're not traditional elastic fiber or collagen fibrillar genes. When we looked at the Hallmark pathways, this is a Venn diagram of differentially regulated pathways in wild type versus knockout, again, for the three models. So elastin's over here in green, LOX is here in purple, and then fibrillin 4 is here in the orange. So unlike the individual genes, which were kind of evenly split between the three models, whether they were unique to that model or overlapping, you can see here that there's a whole set of Hallmark pathways that are very specific to just fibrillin 4 and lysyl oxidase, or to those individual knockouts. For elastin, there's nothing that's specific to the elastin knockout alone. Xenobiotic metabolism is shared between fibrillin 4 and elastin, and then these four pathways here are common to all three of the models. The Hallmark pathways have been curated to eliminate overlap between different gene sets and to highlight coordinately regulated genes. And three of those four Hallmark gene sets, so the IL-2 STAT5 signaling, the inflammatory response, and the coagulation, they have high representation of integrin genes, specifically integrin beta 3, and matrix genes, specifically thrombosponin 1 and some of the fibrinogen subunits in the leading edge. And the leading edge genes are the ones that contribute the most to enrichment in those gene sets. And then that estrogen response at the early leading edge genes have been implicated in GBCR signaling and inflammatory cell phenotype, but they also give a hint that there may be some sex differences linked to the response to elastic fiber defects. And then, as I pointed out in the Venn diagram, there's a large number of common pathways for the fibrillin 4 and the LOX models only, which may be linked to the aneurysm formation in those two models. We also looked at some canonical pathways. So the canonical pathways in MSIG-DB include the KEG pathways, the reactome pathways, the PID pathways, and then a whole host of others. I think there's 1,300 pathways included in the canonical results. And we had, I think, 15 that were differentially regulated between wild type and knockout that were common to the three models. And they can be divided into two groups. ECM associated, which includes the NAVBO collagens, NAVBO core matrosome, NAVBO ECM glycoproteins. It includes genes in the syndecan-1 pathway and the syndecan-4 pathway. Includes genes in the integrin cell surface interactions, and then also in the platelet aggregation plug formation. And so the leading edge genes in all of these groups that are contributing to enrichment have a high number of ECM-associated proteins, but again, not traditional elastic fiber or fibrillin collagens. The other group is integrin-associated. So out of all of these pathways, the KEG ARVC pathway, KEG ECM receptor interaction, leukocyte transendothelial migration, and then these different integrins, all of these have leading edge genes that are integrins. And so those are what's contributing to the enrichment of those. So what we found in this very beginning of our gene analysis summary or analysis is that when we are missing any of these three genes, we have differential expression of ECM and integrin genes, but that the upregulated ECM genes are not necessarily associated with mature matrix, but with provisional matrix observed in wound healing and clot formation. And that the upregulated integrin genes suggest inflammatory and vascular remodeling cascades in response to mechanical stimuli or to the loss of connections between the integrins and elastic fiber components. And so we next wanted to know if these gene expressions are relevant for treating elastic fiber disease. And while we haven't followed up on any of the changes, we have done some work on a model of aneurysms. that are associated with fibrillin 4. And so this is a human with a fibrillin 4 mutation with a large aortic aneurysm here in the ascending aorta. And the fibrillin 4 mice that have a smooth muscle cell specific knockout of the fibrillin 4 gene also have ascending aortic aneurysms. And it's been hypothesized that the loss of fibrillin 4 in the medial layer of the aortic wall leads to mechanical stimuli and or soluble factors that then disrupts this ECM and the smooth muscle cell phenotype and leads to aneurysms. And so we wanted to look at the mechanics of these mice as the aneurysm forms. So we worked with the smooth muscle cell knockout mice, and we looked at various parameters from birth up until postnatal day 30, which is when you can see a definite aneurysm in the SMKO mice. And you can see here by looking at the unloaded diameter that the time where the aneurysm occurs is pretty much between day 7 and day 14, where you have this large dilation. That's also the time when you see significant thickening of the wall. So you see this large jump here between day 7 and day 14 that is then maintained. The aneurysm size also appears to be maintained. So we used our DMT myograph to get pressure and diameter behavior and compare mechanics. So at postnatal day 1, we have the SMKO here in gray and the control here in black. And they're just the same. By postnatal day 7, so before we can detect large dilation in the unloaded state, we do see the dilation in the pressurized state. And then by day 14, you see this large increase in the outer diameter at all pressures, which is then maintained at postnatal day 30. So you can see the diameters here are about the same in the SMKO, where they get larger in the control. So again, we did some calculations to look at material properties rather than just structural properties of the wall. And so if we look now at the stretch versus stress, you can see a slight shift to the left here in the SMKO aorta, which shifts further at postnatal day 7 as this aneurysm is just detectable in the pressurized state. But then as the aneurysm expands, now the material properties come back towards normal. And so we think that some of the aneurysm expansion between these two days may be related to the remodeling that's trying to bring the material properties back towards normal. And then there may be an overcompensation here where it becomes, now it becomes more stiff in this in the SMKO as we get to higher pressures. So we wanted to look at the physiologic loading conditions. So we need to know the pressures across these various ages. And we used pressures that we'd previously found in both wild type and Elastin heterozygous mice. We've shown before that the pressure in a control and an SMKO is about the same. And so we used the physiologic pressures from these ages, and then we went to our pressure diameter curves to figure out the exact diameter that's applicable to that physiologic pressure for each age. So now if you look at the stretch at physiologic pressure, you can see that it's higher always here in the SMKO. The circumferential stress is also higher. And again, these differences start to appear at day 7 and at day 7, so right before the aneurysm really expands, and it may be part of the triggering for that aneurysm expansion. We looked at 2 different stiffness values, either the structural stiffness or the material stiffness, which can be calculated by knowing the dimensions and the applied pressures. And we looked, in this case, the structural stiffness starts out low, which you might expect for defects in elastic fibers, but then through remodeling, the structural stiffness is greatly increased in the SMKO. And then the same thing happens with the material stiffness. And so we've documented these changes in mechanics. We wanted to look at the wall. This is a control at postnatal day 1, and an SMKO, the black, are the elastic fibers. And you can see, you can't tell any differences at postnatal day 1, by postnatal day 7, we start to see these breaks in the elastic fibers and a wall thickening. And then it becomes worse at P14 and P30. And remember that this is the jump where a large amount of aneurysm expansion occurs. So we have these changes in mechanics, we have these changes in elastic fibers, but does it at all relate to changes in cell signaling that might be consistent with some of our results from the knockout models? And so we went back to some older literature from our collaborator, Hiromi Yanagasawa, and she had demonstrated that at P7, an SMKO aorta looks approximately normal, and that you see this aneurysmal dilation by P14. And that's also when, by Western blot, she sees a large increase in thrombosponin 1 protein, which is also one of the 3 genes that was upregulated in all of our 3 knockout models with elastic fiber defects. So it correlates with the aneurysm development, and then it also correlates with regression, where here's an SMKO animal with an aortic aneurysm. If you treat it with losartan, the aneurysm does not form, and it reverses this large increase in thrombosponin protein that you see in the SMKO. So from our studies on the fibulin 4 smooth muscle cell knockout mice, we think that improperly assembled elastic fibers lead to changes in mechanical stimuli, including decreased structural stiffness, which causes remodeling and then leads to additional structural and material alterations. And by P14, these mechanical stimuli lead to an increase in thrombosponin 1, which further exacerbates the remodeling and causes this large aortic aneurysm visible by P14. By P30, the remodeling appears to stabilize, but now we've got this cellular response to these chronic elevations in material and structural stiffness, thrombosponin levels, which may play a role in eventual aneurysm dissection and failure. And so I would like to leave you with one statement, which is that we believe that severe elastic fiber defects in mice lead to common changes in aortic mechanical behavior and gene expression that may provide clues to vascular mechanotransduction in development and disease. And I would like to thank my lab members who actually did all of the work, my collaborators, Hiromi Yanagasawa and Bob Meekum, and also funding by the NIH. And I am ready for any questions.
Okay, thank you very much, Jessica. I hope you found this information beneficial. And now we're going to open for questions from Austin in Jessica's lab. Thank you.
Hello. Okay, so we have a few questions, so I'll just ask them in the order that we received them. The first, can you expand a little bit more on the role of fibulin 4 and 5? Are they scaffolds, enzymatic, or are they part of the fiber structurally?
So they are located, they're associated with the fiber. We know that much. And then if I go all the way back to the assembly process, they have several different hypothesized roles at the coacervation and maturation stage. So fibulin 5 actually encourages tropoelastin coacervation, but discourages maturation. And then there's some implications that it might be involved in this step in moving the elastin to the microfibrils. And then for fibulin 4, it's also been associated with decreasing maturation. Fibulin 4 appears to interact with lysyl oxidase, so it may play a role in the cross-linking. And then fibulin 4 also appears to interact with the microfibers. So they're part of the assembly physically, as well as play a role in how it all gets put together.
Okay, and the next question, are there elastic fibers in other blood vessels besides the aortic blood vessels, for example, the venous and the lymphatic vessels?
Yes, so they are definitely in the veins. so the elastic fibers, let's see, where's this picture? So this is a large elastic artery with these multiple layers of elastic fibers. If we were looking at like a muscular artery, you would see this inner layer. So it's called the IEL, or the inner elastic lamina. And then you'd see an outer one in many vessels, but then none in between. And then in a vein, I believe there's at least one layer, but it may not be as well organized as it is as even a muscular artery. And then I actually don't know what they look like in lymphatics. I believe there's elastin there, but I don't know exactly how it's organized.
Okay, and in the mice that you've studied, have you looked at the cardiac output and stroke volume?
So not in, well, in some of them. In our elastin knockout newborn mice, we did look at that, and it's been published. I don't remember. I think it was slightly decreased in the elastin knockouts. We've looked at it quite a bit in our elastin heterozygous mice that we study through adulthood. And in that case, the cardiac output wasn't different. So just 50% of elastin does not appear to affect cardiac output.
Do you expect that the increased expression of the extracellular matrix proteins that you see in integrins are a part of some adaptive response?
Yes. So I know that all of the matrix we see changing and the integrins we see changing are consistent for the 3 models, and they're happening at embryonic day 17.5 before we see large changes in vessel remodeling, and they're happening in both the ascending and the descending. So they're happening whether we get aneurysms or stenoses or not much change at all. So we think that those changes are an adaptive response to the cells when these elastic fibers are not formed properly. And just in our cursory examination, it seems almost like a wound response, that somehow the cells are interpreting this as a vascular wall injury and trying to form a clot and a provisional matrix.
Okay, and in the elastin deficient and elastin fragmented mouse models, do you also witness increased proteoglycan content in the aorta in addition to smooth muscle cell proliferation?
Yeah, so in our knockout mice, I don't know the answer. So we didn't see the proteoglycans coming up a lot in the gene expression. We did see that increase in the syndecan1 and syndecan4 pathways. And then we haven't done any staining for proteoglycans in our newborn mice. In the fibulin 4 smooth muscle cell knockout, we have done proteoglycan staining, and there is, I don't have the pictures, but if we go look at the elastic fiber pictures, right? So all of the yellow area that's not being stained by elastin, some of this is showing an increase in proteoglycan staining by histology.
Okay, and then I have another question. What are the remodeling responses at P7 that attempt to correct for structural defects?
So, I don't know, because if we look at some of these examples with the thrombospondin, you don't see that thrombospondin increase till P14. But we do see actually there's EGR1 is up before thrombospondin, and that one's, it's not shown here. So it might be something upstream of thrombospondin that's occurring earlier, like EGR1 in this example.
Okay, so that's all of the written questions that we have.
Okay, thank you, Austin.
Would anybody like to be unmuted so you could ask a question in person? Please just raise your hand on your control panel. It looks like Timothy Mead may have a question. Do you see that, Austin?
I think we already addressed his question.
Oh, okay. Any others? Okay, well, great. Thank you very much for joining us. And make sure you join us again for our next webinar on May 3rd for a presentation from Chris Hughes of the University of California at Irvine on a perfused blood-brain barrier on a chip. And just a note, please fill out your evaluation form at the end of the webinar or when you receive it in a subsequent email, and let us know of any questions or topics you might want to see in a NAVBO webinar presentation. Thanks so much, and we'll see you in May. Hey. Bye-bye. Thank you.
Thank you.